You're at the White House pretty much daily doing these briefings. Yesterday's briefing went about 2.5 hours long. I've had a lot of people ask me this question, and so I want to ask you, is this the best use of your time to be up there for 2.5 hours almost every day?
Well, I have to go by the Fauci mantra. I'm always honest. So being there to be able to answer the questions of the media, I think is really important because we're going through a real public health crisis in our country. And I always feel that the media, who I have a great deal of respect for, is really important to be able to answer their reasonable questions, and they are all reasonable questions. The downtime that we have there is really draining. You know, you wait around for it to start, you prepare for it to start, you stand there, you get up. Like last night, I got home at, I don't know, you know what the time was. It must have been about 9 o'clock. And then I had to do C-SPAN immediately after that. And then I had, you know, my usual 600 emails to look at. If I had been able to just make a few comments and then go to work, that would have really been much better. It's the downtime. It isn't the idea of being there and answering questions, which I really think is important for the American public. It's the amount of time per contribution you make per given question. Yeah, so the answer is that I wish I had more time to be able, I mean, we're right now in a very important, what's the right word, process of trying to develop a vaccine, of trying to get monoclonal antibodies. And I go in every morning and I meet with my team at NIAID. But a lot of times they need me for decisions. I make myself pretty available, but that gets you to like literally a 19-hour day, which after a while, you know, it's not easy.
When do you expect to have any preliminary results from the phase 1, and how quickly do you hope to start moving into the phase 2?
Okay, so the phase 1 has 3 components to it, 3 separate doses. We've been through the lower dose, 25, the second highest, 100, and we're entering into the 250. Thus far, nothing, no red flags at all vis-a-vis immediate reaction type safety. One of the important issues that many people don't appreciate, particularly regarding these types of virus, is the possibility of what we call enhancement. Namely, you give a vaccine, you have a somewhat inadequate response, which paradoxically enhances the virulence of the virus. We've seen that with RSV, we've seen that with dengue, and we really want to make sure that doesn't happen now. So what will happen is that we're going into animal studies for 2 reasons. One, to see the immunogenicity. And thus far, in the doses that we've given, it induces a response in a mouse model that is I don't like to be optimistic about this in the sense of giving any false hope, but it's an unusual model because it doesn't cause illness in the mouse, but it allows the virus to replicate in the lung of the mouse, the model. So with vaccinating the mice, the amount of replication of the virus in the lung is dramatically decreased at doses that we think are good. So, you know, remember, be careful of mice because mice often fool you. So the bottom line of that, a little bit more long-winded than I usually say, is that so far, so good in both the animal and the phase 1. What we will likely do is go into a phase 2 sometime in late spring, early summer. I would think sometime probably around June. Now, maybe the second week or so, it's tough to say. What we're going to do, and I'm using this vaccine as kind of a prototype, but there are 2 or 3 others that are also either in phase 1 or getting ready to go into phase 1. And I think the timetable is going to be probably the same. The reason this is going to be interesting and a little different is that given the urgency of the situation, we're going to try and design a phase 1 right into an advanced phase 2A, B, even 3. So if we get, which I think we will, the beginning of a phase 2 sometime in the late spring, early summer, and there are cases around, either in the United States, which I hope not, but there very well may be, but certainly in other of the sites where we have clinical trial sites, we may get an early efficacy signal by the end of the fall, early winter. If we do, then I said a year and a half, we may have something. It still may be a year to a year and a half, but it could be that if there are enough people who have gotten infected and may be protected, that we could have a vaccine towards the part of the mid to late winter of next season. Now, please let me say this caveat. That is assuming that it's effective. See, that's the big if. I mean, people think that, oh, you get a vaccine, it's only a timetable. It isn't just the timetable. It's got to be effective and it's got to be safe. So I know every time I give a timetable to people, immediately the news is we're going to have an effective vaccine on this date. And then when we don't have it, they say, but you said we'd have it. I don't know if it's going to be effective. I hope it is, but I don't know. Anyway, the timetable, bottom line, it looks good.
So let me ask another question that sort of leads up to the when we reopen things. What are you looking at for antibody levels? Everybody's, focused on these new serology tests to say if they've been exposed, but what are you thinking is going to be, the way to tell that someone had enough exposure that they can go back to work?
Well, you just asked 2 or 3 questions that remain open questions. Everyone thinks because antibody tests for so many of the other viruses that we deal with are simple, easy to perform, and give you a lot of information. We're not there yet with this. First of all, there may be one at the most, and we're not even sure of that. Antibody test has actually been validated by the FDA and the NIH. So we're collaborating with them to validate it. So many countries, not many, that's hyperbole, a few countries have taken antibody tests and found out after buying millions of them that they actually don't work. So I give that as a caveat because we need to get a test that works and that's reproducible. When we do, here are the open questions related to what you asked me. A, what is the correlate of immunity? Is it antibody in general, a particular type of an antibody against a particular component of the virus? Question number 1. Number 2, what is the relationship between the titer of the antibody and the degree of protection? Number 3, what is duration of the antibody? Does the antibody effective over 6 months, a year, 2 years? Those are the questions we don't know. So obviously, when we do get these antibody tests that would be more widely distributed, we're going to have to find out the answer to all of those questions. And they're really important questions because the one thing you don't want to do, and I know people are anxious to say, well, we'll give you a passport that says you're antibody positive, you can go to work, and you're protected. The worst possibility that would happen is if we're actually wrong about that. And we say, okay, you're protected. People go back to work thinking that they're protected, do not really adhere as closely to the kinds of things that will prevent you from getting infected, and they wind up getting infected. So we really need to know precisely what we're talking about when we start dealing with antibodies.
There's some discussion about May 1st is a date for some potential reopening, at least in some locations. Is that realistic? And are there locations that you've identified where you could see that happening as early as May 1st?
The answer is it's possible. But as I have said and will continue to say, obviously our mitigation is critical in this tug of war with the virus. But ultimately, the virus is going to determine when we really can safely reopen, not only in general, but in a particular location. So, you see those curves that are shown all the time at the press conference. New York goes way up. New Jersey is way up. Now we're starting to see Boston and DC doing that. Then there's a whole cluster of them on the bottom of communities, states, cities, where there's a degree of infection, but it hasn't done that exponential increase yet. I think how you reopen, if you want to use that word, the economy in those communities is going to depend a lot on the ability to contain what we know will happen. I'll guarantee you, once you start pulling back, there will be infections. It's how you deal with the infection that's going to count. That's sort of a complicated answer because it's a big country with a lot of different dynamic curves to the virus. It isn't one unidimensional curve for the whole country, by no means.
But it sounds like you're saying we should prepare people for the fact that even if May 1st comes and a couple of places are moving toward reopening, people should not expect that the whole country is essentially open for business as usual.
I think you've made a correct statement. I think that right now it's April 14th, 15th, 14th. I think if we are assuming that 2 weeks from now, that all the curves are going to be down, I think that's a bit overly optimistic.
Is a second wave inevitable?
It's not inevitable, but watching what this virus is doing, and the somewhat really disturbing efficiency of transmissibility, I would be so surprised that it all of a sudden disappears the way SARS did in 2002, 2003. So it depends what you mean by wave. If you mean it goes way down and then come September, October, November, we have another peak, I have to say I would not be surprised. I would hope that if and when that occurs, that we jump all over it in a much, much more effective way than we have in these past few months.